here we have Dr. Thomas Braun, our esteemed keynote speaker from the Director of Max Planck Institute for Heart and Lung Research. Sir, my first question is, artificial intelligence has been widely used in cancer treatment and prediction. So what is your take on using artificial intelligence in diagnosis?
I mean, for pattern recognition in images, also for selection of genes and complex sequencing or genome data, epigenetic analysis, I mean, that will be, of course, a tool of the future. I mean, we are already using that to identify in complex RNA-seq or cell RNA-seq data sets to identify gene patterns. This will, of course, in the future further progress, also prediction of structural information, very important. We just talked about this enzyme CPT1B, that we try to identify inhibitors. And for that, of course, structural information, prediction of structural motives, also cryo-EM analysis, so structural information, how different ligands are bound. I mean, artificial intelligence is there already part of the working procedure, and I think this will further develop and further mature. I also have to say there are a couple of limitations. I mean, artificial intelligence, I mean, it's called intelligence, but essentially it's a statistical approach, that simply the computer calculates the most probable outcome based on training data, meaning that something completely new we released by such approaches. It's always an approximation trying to define situations that come closest to events that have already been recorded in the past. And for that purposes, AI is a wonderful tool, but something completely unexpected that will be missed. The human brain is able to find that. But maybe AI gets even better and such issues will be solved.
Thank you, sir. Sir, during the COVID pandemic era, we all face a number of problems and difficulties. So how has it affected your research in a positive impact or a negative impact?
Well, I mean, might sound a little bit theoretical, but I personally kind of benefited, because I had a lot of duties, also traveling around, attending meetings, and it's all disappeared during this 2 years of Corona pandemic. So I could really focus on my stuff without too many distractions. That's the positive stuff. The negative, of course, I think we had not really a lockdown at our institute. We had a lot of measures to cope with the situation, but I think psychologically, that was really also for a lot of young people, also for researchers from abroad, a major issue. They couldn't travel back home. They didn't have contact within their peer group to the extent they had before, so they were rather isolated. Into a new country to work there, postdoc students, then you need these interactions to stay viable. And that was for them, I think, really major stress.
So do you have any suggestions if we are faced with such a pandemic? So what can the research community do to manage the situation more?
I think one has to establish flexible structures.
We have had asked a question about how can plant research be used with cardiac remodeling. So we have ancient knowledge which we have been following from millions of years, where some plants, some medicinal plants, a composition of some medicinal plants can be used for cardiac regeneration. It's proved that it has better heart functioning, it enhances heart functioning. So do we think a possibility of collaboration or working with these ancient plants in this region?
Again, I think I'm quite serious about that. I mean, to really even go down to the biochemical molecular mechanisms. There are, of course, compounds. And I mean, classical drug, which is now a little bit obsolete, digitalis, which changes the calcium homeostasis in cardiomyocytes, that increases contractility. I mean, this is a compound from the plant. It was actually also initially based on the knowledge that this particular plant has this impact initially isolated. And I think there's, of course, a lot of promise in such studies to identify active compounds. I mean, in plant preparations, concentrations, all.
There are a lot of identified concentrations also, sir.
Well, yeah, I think the issue is that depending on how you prepare, how the plant grows, concentrations differ, and then you might have also different fertility efficiency. So I think it's necessary to eventually identify the active compounds, get a proper pharmacokinetics analysis, just for plant extracts. It's skeptical.
Okay, so lastly, how was your experience here, sir? How did you enjoy all of it?
Yeah, I really enjoy it. Yeah, so yesterday evening with the functional editor, I was really amazed that there's so much talent here in the student body. Yeah, I really enjoy people here, seeing all the enthusiastic students and colleagues. Great experience for me.
Thank you so much for your time, sir.
Thank you.
Thank you.